Let's have a look at this hypothetical genotype for a dihybrid cross, where this individual is heterozygous for the one trait and the other trait. So remember that these are two random traits. I don't know what they stand for, maybe tall and green or whatever. But we need to remind ourselves this individual will have a big T and a small t as the two alleles. And now an allele is alternate forms of a gene that are found on homologous chromosomes. So if this is the gene for height, this individual has a big T allele or a tall allele and a short allele for height on the homologous chromosomes. Same thing for color, for example. It will have a big G and a small g on maybe different homologous chromosomes, or maybe the same. But for simplicity's sake, I'm showing it like this. The hardest part for me about a dihybrid cross is determining the gametes. So I'm making a little video to show you why we end up with the combination of gametes that we get. So just to remind you, these homologous chromosomes will both undergo DNA replication and form double-stranded chromosomes. So that pair will form two double-stranded chromosomes, and this pair will also have two double-stranded chromosomes. It's still only one pair and one pair. So it's still only two chromosomes in each pair, but they're double-stranded. So you end up with that chromosome having two copies of the allele for tallness, for example, and that one has two copies of the allele for shortness. Same for the color over here. Now, these chromosomes enter meiosis, and I'm only showing you metaphase and anaphase because those are the relevant phases. So in meiosis 1, we have our homologous pair that has the genes for height, for example, or the T genes, let's maybe call them. And we have our homologous pair that has the G genes, we can call them. They line up at the equator. And at the end of anaphase, these chromosomes start to move to opposite poles. So as these chromosomes are separating, remember the point of meiosis 1 is to separate homologous chromosomes. So one member of the pair goes to the left, and this member of the other pair goes to the left. The other members of the pairs go to the right. So this is random. These two chromosomes could have arranged themselves in a different way. This one could have been on that side, and that one could have been on that side. And that means, as you can see, you're getting the small t gene going this side and the big T G gene going this side. Big T going this side or small g going that side. But I'm going to keep it the way I had it in the beginning, where the small t is going to the right and the big T is going to the left. Now, if we complete this process, you will see that there's going to be two chromosomes in the one cell and two in the other, but only the one chromosome having the T genes and one chromosome having G genes, whereas earlier there were two chromosomes with T genes and two with G genes. So here we separate homologous chromosomes. So let's zoom in on meiosis 2, focusing on metaphase 2 and anaphase 2. So as you can see, we've got our two cells. This chromosome has the two big T's and that one has the two big G's. This cell here has the two small T's and this one has the two small G's. Now, remember, meiosis 2 is about separating sister chromatids. So this is a double-stranded chromosome. We need to make it single-stranded. So when these chromatids separate, you're going to end up with the small t allele going that side, small t going that side, small g going that side, small g going that side. And if I just draw a dotted line down the middle of the cell, you can see that the resulting alleles that are found in these gametes that are going to be produced, this cell will have a small t, small g, and this cell will have a small t and a small g. Same for this cell on the other side, except for the fact that there's going to be a big T and a big G, and a big T and a big G. Now, if there was one thing different, like for example, in the previous step in meiosis, I said these two could have been switched around, which is very possible. So if that was the case, if I just switch around the genes that were in these chromosomes after meiosis 1, for example, so there were small t's there and big T's there, you'll see that before, the gametes produced would have had a big T and a big G, but I've changed the T combination. So now the gametes will have a small t and a big G, because this would be a small t, that would be a big G. Same on this side, small t, big G. So again, the option there would be small t, big G. This side will be big T, small g, and this side will also now be big T, small g. So if you look at the options, the different options for gametes, we have the dominant trait for both traits, big T and a big G. You can have recessive for both, small t, small g. You can have dominant for one, recessive for the other, and then the other way around. So there are 4 options of gamete combinations when we're looking at a dihybrid cross. And it's all got to do with the way the chromosomes line up at the equator. So to simplify that process, all we actually have to do is this. You go back to your original genotype and you just combine a T and a G. Bearing in mind this picture from above, you only end up with 1 T and 1 G. You can never have 2 T's together because those chromosomes are splitting, because gametes are forming. So that big T could either be in a gamete with a big G, or it could be in a gamete with a small g. The other T, the smaller t, could be in a gamete with a big G, or it could be in a gamete with a small g. Now, you saw in the previous example of anaphase that there were multiple options, like there were 2 small tg, 2 small tgs over there, but I just write it once. You would never be asked to explain how you get to the gametes. I was just showing you this video to show you how we end up with 1 T and 1 G in each of the gametes, and how we can end up with a big T being with a small g, or if any of these chromosomes were switched around, like they had lined up differently at the equator, or even crossing over it maybe occurred, and instead of a small t being here, there was a big T that was in that cell, then you could get a big T with a small g. But stick to this method over here, it will not fail you, because as long as you know there's only 1 of each letter in the gametes, because a genotype of an individual has 2 of each letter.